Welcome, everyone. My name is Penny Melquist. I work at InVivo Biosystems in business development, but I'm formally trained as a scientist. So some of you have talked to me in a science role and business development. But today on 17 Minutes of Science, we're going to be talking to Dr. Janis Weeks. Welcome, Janis. And our topic is going to be comparing platforms for testing anthelmintic drugs in C. elegans. So this will be great. You can talk to us about your collaborations with Bayer. But first, I wanted to do a quick bio about you, Janis. Janis got her BS in Applied Life Sciences from MIT. She did her PhD in Biology and Neuroscience at UC San Diego. Your postdoc at University of Washington. You were a faculty member at UC Berkeley. And in 1988, you moved your lab to the U of O. Yes, lucky us from the U of O as a full professor to focus on InVivo Biosystems. So we're very fortunate. And your research, so you have always worked with invertebrate species, leeches, insects, nematodes, used primarily in electrophysiology. You've done a lot of work on the screen ship system, but you focus on identifying neural circuits for behavior with an emphasis on hormonally mediated synaptic transmission and plasticity. Dr. Janis Weeks and Dr. Sean Lockery, longtime colleagues at the U of O Neuroscience Institute, founded Nemametrics in 2011 as a spinoff from the University of Oregon. And another interesting fact about Janis is she's taught neuroscience in Africa for about 25 years with their interest in neglected tropical diseases, specifically helminth infections. And you also, I never knew this about you, Janis, you got funding from the Bill and Melinda Gates Foundation. So you were working on C. elegans, but it was a nice complement to focus on parasitic nematodes. And your current research, aside from all of the other things that you do for us, is to focus on new technologies and platforms to advance research on anthelmintic drugs for animals and humans. And first and foremost, I really have to say, it's an honor to interview you today. I see you on periodically, and we have meetings together, but I really appreciate all that you do for our company and our colleagues, serving as a mentor and what you do around the globe. So thanks, Janis.
Well, thank you, Penny.
All right, so I'm going to start our timer, 17 minutes, and it's probably going to go by in about 17 seconds, but let's go ahead and get started. So why don't we talk about, if you could, sort of talk to us about what are anthelmintic drugs?
Great. Well, again, thanks, Penny, and I'm so honored. This is the first time you've done an interview, and I'm so honored that I'm the lucky one to be interviewed by you. So yeah, let me start by explaining what anthelmintic drugs are. So helminths, the term helminth means parasitic worms of different types. So anthelmintic or anthelmintic just means drugs that kill worms. So the worms are often, they're parasitic, and these various chemical classes of drugs, but in all cases, they're treatments for animals or humans to rid them of parasitic worms.
Mm-hmm. Oh, interesting. So when you think about, C. elegans, a non-parasitic nematode, and parasitic nematodes, in your studies, you use C. elegans. So maybe you could elaborate a little bit more about, not using parasitic worms, but using C. elegans, how that parallels and joins together.
Yeah, that's a really active question in the research community now. So one thing about parasitic nematodes, and I have worked on parasites, so hookworms and ascaris, and those and other mammalian parasites have to be generally raised in mammals. And so, for example, the hookworms I worked on, which infect humans, they can be raised in the lab in hamsters. But in general, it's really expensive and complicated to raise mammalian parasites in the lab. You need to have mammalian hosts, and it's expensive and complicated. On the other hand, those are the worms we want to kill. So working on C. elegans, there's certain, actually high degree of genetic homology in the genomes of C. elegans and the parasites. So sometimes if you have a gene targeted by a drug, it's also present in C. elegans. Or, and this is something we do at InVivo Biosystems, we can insert parasite genes into C. elegans to study them. But anyway, so there's C. elegans is valuable in a lot of ways. For example, finding hypotheses about potential new drugs or how they work. But then they can be followed up in parasitic species. So the field kind of moves ahead by C. elegans as a simpler, more tractable system than the actual parasites.
Sort of like a precursor.
Yeah.
Okay. And give you an indication of kind of what you might see. And I know that for a long time you have worked with as InVivo Biosystems with Bayer Animal Health in Germany on a lot of the similar areas. Could you maybe elaborate on what you were working on and kind of what your hypothesis was?
Yeah, well, this dates back. So Bayer Animal Health in Mannheim, Germany, actually was acquired by Elanco last year. So they're now out of the Bayer family, I guess you would say. But for many years now, I've been meeting up with a Bayer scientist, Daniel Kulke, who was in Germany, but we would meet at conferences and so on. And we eventually, so over the years, and Daniel and his colleague, Iring Heisler, and Daniel came to Eugene and met with us and saw our experimental platforms. Then I spent some time in Germany visiting them and talking. And eventually we came up with this collaboration, which had to do with comparing 3 platforms that are available to test and characterize anthelmintic drugs. 2 of them are electrophysiological, which is my specialty. And then one of them is a platform to measure motility. And just, I'm going to have to explain this at some point, so I will now. So the electrophysiological methods, and these were experiments done on C. elegans. So when Sean Lockery and I founded the company, it was based on a microfluidic device that could record electropharyngiograms, that is the electrical signals from the pumping of the pharynx. And the original device was an 8-channel chip where you could record EPGs or electropharyngiograms from 8 worms at a time. That rig is available at InVivo Biosystems for work that we can do for others, but it's not commercially available. But then the other 2 platforms we looked at in this study are the screen chip, which is a 1-channel EPG device, recording device that is that we do make at IVB. And then the other platform is called the W microtracker, which is a multi-well format device that measures worm motility by when the worms break a little microbeam by moving, it's counted. So what the project ended up being was to compare these 3 platforms to see which are best adapted for which kind of experiments. And Bayer had the screen chip and microtracker in their labs there. And then a postdoc, Stefan Hanel, came to Eugene and did experiments with us for a few weeks using the 8-channel. So that was the project.
Oh, nice. And is it still ongoing or is it wrapping up? I'm trying to remember. I know your paper.
Yeah, the project, we finished all the experiments and the paper, we're returning it with revisions at the end of this week in International Journal of Parasitology, Drugs and Drug Resistance. So anyway, that summarizes all the work that came out of this collaboration.
Yeah, nice. So I'm curious, was there anything that really surprised you out of all of the work that you did?
Well, I'd have to get into some of the results. Surprise. I guess one of the main findings we found, I mean, many people use motility. I mean, there's standard assays people use to look for drugs that will kill worms. You know, they can look at development or reproduction or motility. But electrophysiology can be especially useful because most of the drugs out there act on ion channels. So an electrophysiological readout, it's going to be lower throughput, but often provides you more specific info. So one thing that surprised me and pleased us about our results is that we found that electrophysiological recordings could illustrate drugs or target specific phenotypes. So we found that some electrophysiological signatures could separate this class of anthelmintic drug, which acts on one ion channel, from another class that we studied. And so that was really gratifying. And I don't know if it's surprising. It's what we hoped for, but we were pleased.
Yeah, well, that's good, because it's really never good to be surprised when you're doing research.
No,
unless it's a positive surprise. So it's interesting you think about, using the two platforms, although they're very different, and one doesn't sound like one really favored the other, but they nicely complemented themselves to sort of give more of a fuller picture of what was going on.
Yeah, one outcome of the paper, and actually the reviewers asked us to add a table of this, is, we identified strengths and limitations of all three platforms so that investigators can now pick, what best meets their needs. So that was, a valuable thing for us to find out for ourselves, but also for anybody that wants to use these methods.
Nice. So the next steps, the paper is being, reviewed and edited. What about, any further studies that come out of it that you can elaborate on?
Well, we're not, unless we don't have any plans to continue this collaboration with Bear Animal Health, although we would be happy to, but anyway, that's not in the works right now. But going forward, we're just a lot more confident about, both in-house for us, but also for people out there that have microtrackers or screen chips, what aspects of them are good for which sort of experiment, depending on what you want to find out. And then again, this also sets us up for more the 8-channel EPG work that we can do in-house in terms of when is it most appropriate as a tool based on what you want to accomplish.
Yeah, it definitely seems like, I know you've given talks about this, but the interest in research that's being carried on, especially with funding from the Bill and Melinda Gates Foundation, the research into the parasitic world is really expanding. Wouldn't you say that's an accurate assessment of the past couple of years?
Yeah. A lot of, I'm guessing maybe a lot of the funding, say for international global health, some of it's now, I think, diverted off for COVID, which is, a more acute emergency. But in fact, human helminth infections cause huge, morbidity, not so much death, but just chronic illness and stunning of children. And, there's just a third of the world's population carries a parasitic infection. So it's a huge issue, and it's often been neglected how important this is. But one interesting aspect is that all of the drugs, anti-parasitic drugs, anthelmintic drugs used on humans came from veterinary medicine. So the funding and the motivation to develop drugs for, say, companion animals. If we have dogs or cats, they've been worked as puppies or kittens. Agricultural animals, cattle, goats, horses, all those. There's a huge need for new drugs because of drug resistance, and then the way it works, those then typically set up a way to make those drugs work for humans. So I think there's a lot more investment in animal anthelmintic drug development right now, but that always is a plus for human infections as well.
Yeah. Well, that nicely segues into my other curiosity question. If you could maybe talk about, your work in Africa, working with, parasitic research and educating people in neuroscience. It nicely complements what you do for us at InVivo Biosystems, in the sense you are the Chief Global Health Officer, which I didn't say that earlier, so my apologies, and co-founder. If maybe you could talk about that in our last couple minutes. We have 5 minutes left, so
okay.
You're doing great, yeah.
Yeah, well, I just out of the blue got invited right after the end of apartheid in South Africa. I just got a phone call asking me if I would like to come in and teach in the first neuroscience workshop coming into South Africa after apartheid, and that was in 1996. And that just flipped a switch, really. And so since then, I've been involved with the International Brain Research Organization, which offers research and education capacity building in neuroscience, really all over the world, but especially on low and middle income countries. And I've sort of specialized in Africa. And the last decade or so, a small team of us have focused on a workshop called Teaching Tools, which takes junior, young, beginning faculty members and works on new, innovative, integrative ways to teach neuroscience and neurology. So we've been running that workshop. We didn't run it last year, obviously, because of COVID, but we're thinking about doing it this year. But it was being in Africa year after year for those workshops. And then I'm involved in Zimbabwean music. So I've spent a lot of time in Zimbabwe and just seeing these diseases firsthand. That's what really just changed my research directions, in the mid 2000s, to say, how can I take my neuroscience expertise, and even though what I was doing before I loved, how can I apply it to an applied global health problem like parasitic disease? So that's what got me here.
What do you think your next steps are going to be?
You mean career-wise, research-wise, travel?
Oh, just like Africa, because your whole career in like neuroscience, physiology, parasites, Africa, these are all related, and so it seems like there's another, move after that, and tying all of these things together.
Well, I think for now, and especially with all the uncertainty with the pandemic, I'm just going to keep being, the parasite contact at InVivo, and work with customers and researchers who are interested in anthelmintic drug development or other drug development as well. And then I'm going to keep going to Africa teaching again. That may pick up. That may be possible again starting this fall. I also, even though I'm retired from U of O, I come back and teach one course a year on tropical diseases in Africa for biology students. So I've kept doing that because it's a really great way to, alert our students about all the important research questions and, health problems out there that hopefully they can graduate and go out and help solve.
Yeah, definitely. And I'm curious, are there any like anthelmintic drugs that are, at the top of your mind in terms of popularity, like top 2, top 3?
Well, there's a lot of drugs that are in the benzimidazole class, like albendazole and so on. And those drugs act by destabilizing microtubules. But there's 2 other classes really. And these are ones we used for this study with Bayer Animal Health. One is the ivermectin drugs like ivermectin. You've probably if you have pets, used ivermectin to worm them. So that class of drugs, they act on glutamate gated chloride channels on muscle. And then the other class that we've worked on a lot is drugs that activate acetylcholine receptors, like levamisole. And that's an agonist of ACH receptors. And both of those classes of drugs cause paralysis of worms. And then say for intestinal parasites, once they're paralyzed, they can just be swept out of the digestive tract. So I personally, as an electrophysiologist, favor the drugs that act on, neuromuscular transmission or synaptic targets. But there are other classes out there that act, that perturb other aspects of the worm's physiology.
Nice. Well, we have about 20 seconds left, and I just want to take the time to thank you again for being such an amazing resource. I always am sending you emails and questions about parasitic, questions that I have from other people, and just out of curiosity, you talking about Africa. So thank you so much for being an amazing colleague, a fantastic mentor, and an excellent example of a woman in science. You're amazing.
Please.
And just keep on keeping on, and don't hesitate to use Dr. Weeks. I'm going to use it on you.
Well, thank you.
Yeah, and I want to take 20 seconds to give a shout out to the person that produces and directs and edits and curates this entire series, Hannah Houston. One day you're going to be interviewed, and I'll make sure that that happens. But you know, without Hannah, this would not happen. She does all of the behind-the-scenes work. She's got several screens going right now so that we can live stream this. So thank you, Hannah. Thank you, Dr. Weeks. It's a pleasure to be your colleagues, and tune in next week for another great show, and we'll be back. Thank you.